All right, let's dive into one of the most incredible interactions in all of biology, the antigen-antibody reaction. This isn't just some abstract concept. It's the absolute bedrock of your immune system, and it's the secret engine powering the medical tests we rely on every single day. And that's not an exaggeration. It's literally true. Think about it. A single drop of your blood holds a microscopic diary of your entire life. Every infection you've ever fought off, every vaccine you've ever had, it's all recorded in there. And the language it's written in? Well, that's the language of these antigen-antibody reactions. So let's break it down. How does this molecular handshake, this secret communication, actually work? Let's get into the fundamentals. Okay, at its very core, this whole thing is about specificity. It's like a perfectly cut key for a very specific lock. An antibody doesn't just randomly grab onto an invader or an antigen. No, it's designed to recognize one tiny, unique part of it, a molecular signature we call an epitope. And the bond itself, it's not like superglue. It's made of a bunch of weaker forces that together create this really strong connection, but it's also reversible. It's a handshake, not a handcuff. So how do we talk about how strong that handshake is? Well, we've got two key terms here, affinity and avidity. And the Velcro analogy you see here is perfect. Affinity is like the strength of just one of those tiny hooks binding to one tiny loop. It's a single point of contact. But avidity, that's the real magic. That's the combined strength of the entire strip of Velcro. Since our antibodies have multiple arms to grab on with, avidity gives us a much better sense of the true binding power inside our bodies.
Right, so this whole process happens in two stages. First, you have the primary stage. This is that initial handshake, the moment the antibody first binds to the antigen. It's super fast and totally invisible to us. But then comes the secondary stage, and this is the one we can actually see and measure in the lab. This is where all those individual handshakes lead to a bigger visible effect, like clumping. We call it agglutination. And that visible result, that's the key to so many diagnostic tests.
And that brings us right to the lab bench. How do scientists take this invisible, microscopic interaction and turn it into a powerful tool that can tell you if you're sick?
This graph illustrates a principle that is absolutely crucial to understand. The strongest reaction, the most clumping you can see, only happens in what we call the zone of equivalence. This is the sweet spot. It's where the proportions of antigen and antibody are just right to build a huge interconnected web or lattice. If you have way too much of one or too much of the other, the reaction actually gets weaker and can even disappear. Remember that point? Because it's going to be really important in a minute. This is where things get really interesting. It raises a fascinating puzzle. What happens when an antibody binds perfectly to something, say a red blood cell, but for whatever reason, its other arm can't reach another cell to form a bridge? These are called incomplete or blocking antibodies. They're there, they're doing their job, but they're completely hidden from view. So how on earth do you detect them? The answer is just brilliant. It's called the Coombs test. The secret ingredient is something called Coombs reagent. Get this, it's an anti-human antibody. It's an antibody that is designed to bind to other human antibodies. So you add it to your sample and it acts like a bridge, grabbing onto those invisible antibodies that are already stuck on the cells. It forces them to link up, creates that clumping we were looking for, and bam, the hidden reaction is revealed. This is absolutely critical stuff, especially in blood banking. Now, these fundamental ideas, the specific binding, the visible reactions, they've evolved into an incredible toolkit of modern tests that are way faster, sharper, and more sensitive than ever before. Let's look at two major players. On the left, you have immunofluorescence. It's pretty straightforward. You stick a fluorescent dye on your antibody, and it literally glows under a microscope. Cool, right? But what if the signal is really faint? Well, that's where ELISA on the right is a total game changer. Instead of just a dye, you attach a powerful enzyme to the antibody. This single enzyme can then act like a tiny factory, churning through thousands of chemical reactions to produce a massive, easily measurable color change. It's all about amplification. It lets you detect even the tiniest traces of something. And when you need the final word, the ultimate confirmation, you turn to the Western blot. This is often the test used to confirm diseases like HIV. What it does is genius. First, it separates out all the potential target proteins by their size. Then, it uses antibodies to see exactly which one of those separated proteins your body is actually reacting to. It's that two-factor authentication, size plus antibody specificity, that makes it so incredibly reliable. But here's the twist. Even with all these amazing tools, there's a really surprising diagnostic trap you can fall into. A totally counterintuitive situation where having more of something is actually worse. Seriously, think about that for a second. It just doesn't make any sense, right? You'd think a patient with a super strong immune response, just loaded with antibodies, would light up a diagnostic test like a Christmas tree. But sometimes the test comes back negative. How is that possible? Well, this bizarre situation has a name: the prozone phenomenon.
Let's go back to our trusty graph. Look all the way to the left, in that zone of antibody excess. You see how the reaction curve just completely tanks? It plummets to 0. Even though the sample is flooded with antibodies, the visible reaction we need just vanishes. This is the prozone.
So here's what's actually going on, step by step. First, you have this overwhelming flood of antibody molecules in the sample. They vastly outnumber the antigen particles.
So what happens next? Well, because there are so many antibodies floating around, they just mob the antigens. Every single available binding spot on every single antigen gets occupied by its own individual antibody molecule.
And here is the crucial result. Because every antigen is coded by individual antibodies, no single antibody can grab onto two different antigens to form a bridge. You can't build that big, interconnected lattice network. And without that lattice, there's no visible clumping, no precipitation, and the test looks falsely negative. So how do you fix this? The solution is beautifully simple, but absolutely vital. You just dilute the sample. By performing a serial dilution, you systematically reduce the concentration of antibodies. Eventually, you dilute it enough that the ratio of antibody to antigen falls right back into that perfect zone of equivalence. And suddenly, poof, a strong positive reaction appears. This is so critical for avoiding dangerous false negatives in tests for things like syphilis or brucellosis, where these super high antibody levels are pretty common. So let's bring it all back home. From the diagnostic lab bench right back into our own bodies, these amazing reactions are the foundation of our health, constantly working to reveal the secrets hidden in our blood. Every single second of your life, your antibodies are out there performing these incredible feats. They're neutralizing nasty toxins and viruses before they can do harm. They're physically blocking bacteria and other microbes from latching onto your cells. They're activating other parts of your immune system like the complement cascade, and they're tagging invaders for destruction, a process called opsonization, which is basically like putting a giant eat me sign on a bacterium so your immune cells can find and devour it.
It's just such a complex, elegant, and powerful system. It really makes you stop and wonder, doesn't it? What other incredible biological secrets are humming away inside you at this very moment, silently keeping you safe, just waiting for us to discover them?